My name is Amy Howard, and this is Silei Zhu, we're here from the University of Oxford, and I've recently moved to Imperial. But we're here today really to talk about some work that we've been doing over the past several years in Oxford, and this is acquisition of a data set that's very complementary to BigBrain, and this is the BigMac Dataset. So the BigMac Dataset is a new open access resource combining in vivo MRI, post-mortem MRI, and multi-contrast microscopy in a single whole macaque brain. We've got in vivo MRI acquired over multiple scan sessions, around 270 hours of post-mortem scanning, well over 1,000 hours of microscopy, resulting in around 4 terabytes of raw data. And why bother? Well, I don't think we need to say this within this room, but it's really this common goal of trying to link microscale cellular structures to these macroscale features of whole brain organization. And here our aim was to do that within the same single brain. So we really know we're comparing cellular like for like. So let's unpack BigMac a little. So our in vivo data includes structural MRI. We've got task, FMRI, resting state, and diffusion MRI data. Post-mortem, we've got structural again, T1 maps, and then an extensive diffusion MRI protocol, really to look at both the structure and microstructure of the brain. And having both data sets provides us fantastic opportunity for us to link our post-mortem MRI and microscopy results to the in vivo data within the same animal. The post-mortem diffusion protocol was fairly extensive, so we've got data at 2 different resolutions, 0.6 millimeter with a single shell of B value of 4,000, and then multi-shell data at 1 millimeter, where we've also got one of the highest angular resolution data sets with 1,000 gradient directions in the outer 2 shells. The high spatial resolution is nice because we can characterize the fiber orientations with fewer partial volume effects, whilst the 1 millimeter data allows us to obtain very high SNR when we push to these higher B values, so we've got more diffusion contrast. We also could obtain a quite extensive number of gradient directions in a feasible scan time of just over a week. And further, we've got linear and spherical tensor encoding, which just supports the use of more advanced microstructural models. So what does of course, with a very high B value, we obtain very nice diffusion contrast. So here I'm just showing some of the raw diffusion-weighted images that you can see a bit later in the workshop as we trace this spiral over the Q space sphere. All of our data was acquired on a 7 Tesla small animal scanner using a spin echo multi-slice sequence with single line readout. So this is really nice because we have very few image artifacts, but it is fairly slow, resulting in the long scan time. What does data with 1,000 gradient directions look like? So here I'm showing just a single voxel, the signal in a single voxel, where each point shows the signal along different gradient directions. And we just have the voxel from 2 orthogonal views on the top and bottom. You can see with 1,000 gradients, we can really start to characterize the diffusion signal in great detail. We can see the shape of the signal, and we can even see this kind of slightly smaller bump. And that can provide more microstructural information. But although, this looks beautiful, we wanted to ask, does it really provide any additional information, and particularly when we want to look at fiber orientations and running tractography. And so what we did was we took our 1,000 gradient data set and we divided it into subsets. We fitted fiber orientations using diffusion models, and then we ran tractography. And the kind of high order result was that we found that high angular resolution improved tracking, particularly through crossing fiber regions. And this was because we had more crossing fibers. We found additional secondary and tertiary fiber populations with the increased number of gradient directions. And also we got longer streamlines throughout the brain. And here I'm just going to show an example of a single tract that we reconstructed. So this is the superior longitudinal fasciculus 2. On the top, we have the 1,000 gradient directions. In the middle, we have more typical data with 64 gradient directions. And on the bottom, we have high spatial resolution, because for the same amount of time, do you get lots of directions or high spatial resolution? And what we can see is that with the high angular resolution, particularly for this tract that's going through crossing fiber regions, we're able to get this really nice coherent tract core that follows our anatomical expectations, projecting into the more anterior brain. Whilst in the other 2 data sets, we can see we get a shorter tract core and also this kind of noisy offshoot, which is streamlines jumping onto other tracts. So in BigMac, we have this rich MRI data, both in vivo and post-mortem, but we want to compare it to microscopy data within the same brain, and so what microscopy do we have? After scanning, the entire brain was sectioned along the anterior-posterior axis, where we acquired consecutive slices, and we assigned them to one of 6 microscopy contrasts. So we have polarized light imaging to visualize fiber orientations, a Gallium silver stain to visualize myelin, a Crescel violet stain to visualize Nissl bodies, and then we have 3 sets of sections that are unstained and unassigned and kept for future staining. Each stain is repeated every 350 microns throughout the brain, and we have whole brain coverage. Here I'm just showing some example polarized light images. Of course, here we just utilize the birefringence of myelin to estimate the primary fiber orientation, here with a resolution of around 4 microns per pixel. We don't have a rotating stage or anything, so a tilting stage, sorry. So we primarily can just look at the fiber orientations within the plane, as shown by this 2D color wheel. Of course, we get beautiful orientations within the white matter, but also we can see these nice fanning patterns when we go into the cortex, and we can even see variations across different cortical layers. Another thing of interest, particularly when combining this information with diffusion MRI, is that we can see these beautiful fibers projecting through the deep gray structures, which are typically challenging to get from MRI alone. Our second microscopy contrast is a Gallium silver stain, so this is where the myelin has been stained brown, and we acquire this data with an order of magnitude higher spatial resolution of around 0.28 microns per pixel. We can, of course, use this to, again, review these beautiful fanning patterns across the cortex. We can use this data to look at different subfields of subcortical structures or the hippocampus. And even in the more densely stained white matter, we found these really interesting kind of textures, like these wave-like undulations found in the corpus callosum, which is typically considered a very coherently organized region. With the high resolution, we can also visualize single fibers, particularly where the staining is a bit less dense. More recently, we've moved on to look at the cytoarchitecture as well. So this is our Nissl stain. It's a Crescel violet stain where the Nissl bodies have been stained purple. Of course, this kind of data is often used to look at differences across cortical layers, but due to the high resolution, we can even visualize single cells, such as these small round glial cells in the white matter, as well as identify different types of neurons in the cortex. And I think here, this is where you can really see that by having consecutive slides with different microscopy contrasts, we can build up this kind of multimodal characterization of different structures. So this is the data that we have so far for microscopy, and we have whole brain coverage. Now, PLI is nice because we get a quantitative estimate of fiber orientations out of it, right? But histology is a bit more tricky. We have to work a little bit harder to extract quantitative metrics. And this is particularly because staining densities are often only semi-quantitative, making cross-slide comparisons challenging. So when we see faint staining, it can be difficult to know, is it just because there are no cell bodies, or is it just a staining artifact across the slide? So recently we've been doing some work with an aim to build whole brain quantitative microscopy volumes that can translate high resolution information from histology into MRI space. To do this, we first perform data-driven stain segmentation, and then we combine this with high-quality co-registrations to try and build these atlases of how the tissue microstructure is varying and co-varying across the brain. Stain separation was performed using data-driven thresholds. Here we separate the brown myelin from the background staining using 3 thresholds related to weak, moderate, and strong staining. We then calculate the number of stained pixels over a local neighborhood to calculate the stained area fraction, here over a 40 micron neighborhood. By doing this across the entire section, we can acquire these 3 different stained area fraction maps related to weak, moderate, and strong staining, where the weak threshold is able to better visualize myelin variation across the cortex, whilst the strong threshold can show us myelin variation across the more densely stained white matter and subcortical structures. Now we have these quantitative metrics, we can do nice things like compare them to curvature estimates from FreeSurfer, for example, where we were able to, across the brain, see an interesting difference in myelin across the cortex, with considerably more myelin in the gyral crowns compared to the sulci. We can perform similar stain segmentation using Nissl stains to produce these stained area fraction maps that, again, are kind of a faithful representation of the Nissl variation. But here we can go further and we can aim to segment single cells from the histology data. And once we've done this, we can then extract morphological metrics such as approximate cell area, circularity, the number of cells, the radius, or the nearest neighbor distance. With these histology data, we can also perform structure tensor analysis, which is just a type of texture analysis that allows us to again estimate the primary fiber orientation per pixel. Here I'm just showing it again using the 2D color wheel similar to PLI. And what's nice here is that we can of course do this on a myelin stained slide, but we get very similar information if we also do it from a Nissl stained slide, where both are able to see these interesting details such as these interdigitating fibers entering the corpus callosum. So once we've got our quantitative metrics, to reconstruct them into a microscopy volume, we need to perform high-quality co-registration. And in BigMac, we did that using TIRL. And today, one of the workbooks will allow you to play around with the TIRL warp fields and actually do some of this mapping yourself. In BigMac, we used a two-stage registration process. So in stage one, we took each microscopy slide and separately registered them into the MRI volume. And then in stage two, we optimized over all slides simultaneously to ensure that our slides were parallel and that they were a kind of fixed distance apart. Here's just some example outputs. So on the left is after stage one, where you can kind of see the slides are crisscrossing a bit, whereas on the right, you can see they're now parallel, which is as we would expect from how the brain was sectioned. Combining these co-registrations with our quantitative metrics, we can now reconstruct these 3D microscopy volumes. So here I'm showing an example of the Nissl stained area fraction, where even though each stripe within the coronal plane represents data from different slides, we observe smoothly varying metrics across the brain, which suggests that our slides can be combined with minimal impact from the between slide staining variations. Further, our volumes describe microstructural variations of interest across the white and gray matter, across cortical layers, and across subcortical regions as desired. And of course, we don't have a single microscopy volume, but we can reconstruct them according to all of the different quantitative metrics that we've extracted. So we have metrics related to fiber orientations, related to myelin and cell body density, as well as all of these different morphological metrics. These 3D volumes really allow us to quantify cellular variations then at different scales. So at the macro scale, we can see that we have increased cell body density in the posterior of the brain, in the posterior cortex, should I say, but not in the white matter, meaning that, well, suggesting that this is not an artifact of the slide staining variations across the sample. At the mesoscale, we can quantify cellular distributions across different regions of interest. So here, where we're looking at the cell body radius, we can faithfully see small glial cells in the white matter with a small number of large BET cells in the motor cortex. And then at the microscale, we can take surfaces from FreeSurfer, we can extract cortical profiles stretching from the peel to the surface boundary, and we can then plot our microscopy metrics along these profiles. Here, the kind of profiles faithfully represent variations in the Nissl data, such as this dense staining in layers 4 and 5, which appears to be primarily driven by an increase in the number of cells, as well as these small round glial cells in the white matter, as expected. Together, these profiles can build a microstructural fingerprint with which we may be able to aid cortical boundary definition, for example. So I hope in my kind of whirlwind introduction of BigMac, I've convinced you that BigMac includes rich MRI spanning different tissue states, in vivo and post-mortem, and a wide variety of MRI contrasts from functional to structural to microstructural MRI. Further, we have these quantitative microscopy metrics to characterize the cyto and myeloarchitecture across the brain. And we believe there's a whole host of applications for this kind of whole brain multi-contrast data, from neuroanatomy to multiscale neuroscience, developing MRI methodology and the validation of MRI models, as well as machine learning and data fusion methods, one of which Silei is going to come and talk to you about now.
So next, I'm going to talk about a tool we developed. We try to combine the advantages of diffusion MRI and microscopy and try to study how the brain connects, try to better improve our knowledge about brain connectome. So as we have seen in the BigMac Dataset, microscopy and diffusion MRI have their own advantages and disadvantages, and they provide quite complementary information. So microscopy has very high spatial resolution, but in our data it is in 2D plane. And in comparison, diffusion MRI is in 3D, but the spatial resolution is lower and at a millimeter scale. So in this work, we try to develop a data fusion method, and it can combine the advantages from two modalities. And here is a brief overview of what technique people usually use to study the brain connectivity. And for example, people can use chemical tracers, and we can know the precise mapping of the projection from the injection site. And in addition, the diffusion MRI, by using the tractography, we can get the whole brain mapping of the string lines. And the other technique is the myelin-sensitive microscopy. And for example, the Gallius silver staining we just seen and the polarized light imaging are all sensitive to myelin and can give us the information of the axon architecture in the brain. So we are going to combine two modalities. And so this is the step how we are going to do that. So on the one side, we have diffusion MRI, and usually we can model the fiber orientation in each voxel by some physical modeling. And one common method is called ball and stick model, and it is implemented in FSL. And as its name showed, it modeled the fiber orientation by describing the diffusion signal with a ball compartment and stick compartments. And we can get the crossing fibers and fiber orientation distribution in each voxels. And on the other side, we have the polarized light imaging, and it can provide fiber orientation at a very high spatial resolution. And we have co-registered them together with TIL, we just see. And then we take each microscopy orientations. And to make these two orientations comparable, the first thing we do is we decompose the 3D orientations from diffusion into 2D. So we decompose it into the through-plane orientation and in-plane orientations by projecting onto the microscopy plane. And then we compare the in-plane orientations to the microscopy orientations. We can know which ball and stick sample is most aligned with the microscopy orientations by calculating these angle differences. So we select the most aligned diffusion samples, and we use these samples to inform on the through-plane orientations, which means the microscopy gives us the in-plane orientation, and diffusion might give us the through-plane informations. And in this case, we can get the hybrid orientations that is in 3D and at native resolution of microscopy at 4 micrometer, and this can also retain the rich property of the microscopy. So here is an example slice, and we can see the PLI gives us the orientations shown in the subplot B and A. And the diffusion MI, when we decompose into in-plane and through-plane, we can also get two maps. So we can compare the diffusion in-plane orientation with the PLI orientations. By comparing these angle differences, we can see the values were quite low, which means they align quite well. They are quite consistent. So we then combine the diffusion through plane, which is a subplot D, and to the target PLI orientations, which we can get the 3D orientations. And one benefit of our hybrid orientation is it is very high resolution, and we can reconstruct it into various resolutions. So here we show it can be combined to 0.04 millimeter and all the way to 0.6 or 1 millimeter. So this can help us study different questions. If we want to study how the spatial resolution will affect the tractography, how it will affect the brain connectome, and we can use the same underlying data and reconstruct at various resolutions. And we can also, so maximally, we can construct resolution at 4 micrometer in plane and 350 millimeter through plane. This is limited by the gap between the PLI slices. So the first thing we try to validate is we reconstruct at 0.6. So this is the same as our diffusion MRI data. So these are from the same macaque brain. And so they also have comparable anatomical structures. So we compare with the traditional method, and showing at the 0.6 millimeter, we can capture the reasonable structure, fiber orientations, which is the U shape shown here. And in addition, we can do the tractography. And we are also going to show how we are going to do tractography in the tutorial after the talk. So it is a very simple command running tractography, and we can capture anatomical structures, including the crossing fiber in the corticospinal tracts, and also the backward curving structure in the optic radiation. And we also proceed to construct the white matter tract spanning the whole brain. And here we use the X-tract atlas in FSL. So it includes 42 tracts. So we can provide a more whole brain level microscopy informed orientations. So we are also going to provide these whole brain fiber orientations, and you may be able to play around with these orientations after the talk. So what is the benefit of these microscopy informed orientations? Like what actual information we can learn from it? So we use it to study what diffusion MRI challenge it can solve. The first challenge we look at is the gyro bias problem. So it describes the situation in the cortical gyrus. It means the streamline usually terminates at the gyro crown instead of terminate sharply and curve sharply into the gyro walls. So we can see in the PLI, we can get this beautiful fanning pattern, but in diffusion MRI, because the partial volume effect or limited by the spatial resolution, we usually feel challenging to get it. And in our hybrid orientation, we can see as we increase the spatial resolution, we can overcome this gyro bias problem. In the 1 millimeter, we couldn't see obvious fanning pattern, but in 0.4, we can get this like more even distribution in the gyro crown and gyro walls. And if we do the tractography, we can also see this trend. And in diffusion MRI 1 millimeter, we can observe this problem. But if we increase the resolutions, in diffusion MRI in hybrid orientation, we can overcome this problem. And we can see a more obvious trend in the hybrid orientation as well. And in addition, we also studied a problem called bottleneck problem. And this describes white matter organizations within the brain. And it is prevalent in a lot of brain regions. For example, in the internal capsules shown here. So when different tracts merge in a small regions, sometimes we couldn't distinguish where the tracks go, where the tracks terminate. So this causes a lot of false positive problem in the tractography. So we also use our hybrid microscopy informed orientation to study it. We start the streamlines from the motor cortex. So we see whether the streamline projecting from the motor cortex and into the internal capsules, whether it will retain these anatomically well-known organizations. Different motor cortex have the representative to different body parts, and it will usually have a distribution from anterior to posterior in the internal capsules. And in the diffusion MRI, we can see we see it from different color region on the right, and we can in the internal capsules, these voxels are color coded by the maximum density from the motor cortex, and we can see it merge, and we couldn't distinguish where it is projected from. This is usually we describe as the bottleneck problem. But at the same resolution, this is from the microscopy informed, we can better get these distributions from the anterior to posterior axis. And we can also get similar pattern if we increase the resolutions. And interestingly, if we have 1 millimeter, we can also better resolve these structures with our hybrid orientations. So this means like our microscopy have some more specific information of the fiber orientation that can help us better solve the brain connectivity problem. And this method is also generalizable to other microscopy contrast, like Gallium staining and anisole staining in our data sets. And we also have the online tutorial available, and also the code is openly available. And another applications of the BigMac is we use the deep learning method to solve the brain connectome. And why we are going to do that is, you may realize in the hybrid orientation we work with the post-mortem data, and we didn't talk about in vivo data a lot. And there is a question of how we are translate the post-mortem information and our post-mortem knowledge, microscopy, to the in vivo. And so, why this is a challenging task, and this is the aim of our next project, and the project we're working on, is because the microscopy is the fixed tissue, and the in vivo data is the living tissue. So tissue type is very different, and the data type is also different. The microscopy gives us the orientation as the vector field, but in vivo data give us the diffusion and my signals. So the data type and tissue type are both different. So this is making it challenging. But postmortem diffusion can serve as a crucial intermediate, which is an advantage of BigMac Dataset, because it has the microscopy, postmortem, and in vivo diffusion all in the same brain, and so with a well-aligned brain structure. So this can help us translate information all the way from the microscopy to in vivo diffusion. And we want to use this to inform and better improve the estimation of fiber orientation in the in vivo diffusion data. And we are going to use the deep learning method to solve this. So the network will learn the microscopy informed fiber orientations from diffusion and my signals. And these microscopy informed orientations here is the hybrid orientation we just talked about. In our task, we want to make this network both work for the postmortem diffusion and in vivo diffusion. So we use a structure called domain adaptations. And here domain here means the two domains are sharing the same task, but they have two different data distributions. And in our task, the domains are postmortem diffusion and in vivo diffusion. So the network will first aim to learn domain invariant features, and then it will inform on the microscopy orientations. So to start with, here is a simplified network architecture. We take out the blocks of the diffusion signals, because the fiber orientation is quite local, so we are doing a more local way. And then we will go the data with the convolutional layer and the fully connected layer. We can aim to estimate the fiber orientations from the postmortem diffusion data. And to make it work for the in vivo data, we have an actual component of discriminator. So the discriminator will aim to distinguish between the postmortem and in vivo. And then the first feature extractor will try to confuse the discriminator. So this will make the end layer of the feature extractor to be invariant to postmortem and in vivo data. And we train this network in the BigMac dataset. So we have the postmortem and in vivo in the BigMac. And then we also fine-tune it to work for the human data. So here is an example. We use the UK Biobank dataset. It has a lower spatial resolution of 2 by 2 by 2 millimeter, and it's in vivo, and here we work with the single cell data. And we can see with some usually traditional people use method as like the constrained spherical deconvolutions at the top. We usually get some noise in the gray matter and white matter boundary. But with our network, we can remove these kind of random peaks and better estimate these orientations. And this may be informed by our microscopy specificity. What I was trying to show was disadvantages of the gray matter white matter boundary can help us improve the tractography as well, and we can get beautiful superficial white matter tracks. So this network, we are still working on it, so we are it's not included in our tutorial, but we aim to apply it to macaque and to the human and both postmortem and in vivo dataset. And so we just talk about two applications, and we are also working on some more applications like the subcortical regions of the brain. And Amy will talk through about the tutorial. We are going to share how we are going to use these tools and how we are going to play with the BigMac dataset.
Yeah, thanks for the presentation so far. You showed that the higher resolution helped to overcome the gyral bias in tractography. And you said it's not only tracts do not only terminate in the gyral crown, but also in the walls. But what about the sulcus, the bottom of the sulcus? Is there also more tracts now, or they're still flowing around? Or can you do you have a result on that or some comment?
Yes. Yeah, so we also aim to like have a more quantitative way of seeing whether it's more even across the whole brain. Like this will consider the cell size and whether it will improve as well. And yeah, so in our BigMac dataset, it's improved, but we didn't show here. Yeah.
From the microscopic data, is it known that as many tracts terminate in the sulcus, like at the bottom as in the gyral crown?
Have a look at some of the data today. I mean, so as I was showing the myelin differences across the brain, so you see less myelin in the sulci. So
there is a gyral bias.
I'm not the person on this, but at least according to our data, that's what we see in myelin. That doesn't mean there aren't fibers there. It means that we can't see them, those ones, at least in some of the regions, and it's not everywhere. But yeah, please have a look at some of the data.
Hi, thanks a lot for the talk. It was really nice. My question is regarding the layer profiling stuff that you showed. So if I understand correctly, you obtain the layers using free surfer-based segmentation, right? Mm-hmm. And then you do your profiling based on the cells obtained from histopathology. So when you overlay the layers on top of histology, do you really see that the layers from free surfer actually land up at the correct histology thing?
So it depends a bit on how you create the layers, right? So whether or not you want to have more equivalent volume or equivalent distance across the layers. So they're not, I wouldn't argue, when you get the layers from free surfer, they're not anatomically faithful layers, but what they are is a set of coordinates between which we can compare the MRI and the microscopy really nicely, right, and across the brain. So I would love someone to take this data and take the histology and do proper layers. That would be really cool.
So did you choose between equivolume or equidistance? Which one did you go with?
I can't remember which one I've used here, but I have both of them.
And sorry, one more question. And regarding the convolution neural network, the domain adaptation that you showed, how do you select your patches? Is it across the whole brain or some specific region? Our input is only 3 by 3 by 3 blocks of voxels. So because we only have one subject, so we sample it into a lot of blocks of fiber orientations. Because fiber orientation is quite local, so it may be continuous and it may utilize the neighbor information, but it's not like a whole brain. It needs to be input into network. So yeah, that's how we define the input.
But the blocks come from everywhere in the brain, right?
Yes, the block comes from everywhere of the brain, both gray matter and white matter and subcortex.
Perfect. Thank you.
Thank you very much. That was a really interesting talk. I had a quick question about potential applications. So let's say I have histological white matter staining through a macaque, but it's sparsely sampled. So I have 50 sections for one brain. Is your tool still useful? Are there potential applications that I could use your tool for? Or do I just not have enough data to really make any meaningful assessments about white matter in those specimens?
Which bit? What do you want to do with it? Do you want the registration?
Oh yeah, not the registration. No, the I guess finding fiber orientation in plane.
So you can always do doing fiber orientations. That's just a registration thing, right? But the hybrid, I mean, if it's, depending on how densely sampled it is basically the resolution that you can get to, right? So you could have very anisotropic voxels, but, are they meaningful is a question. You can, interestingly, complement it with diffusion stuff. So, we have a small section in the middle of our brain that we don't have any data from because it was sectioned in 2 halves. and we kind of fill in there with diffusion. But then you're losing the kind of magic of it, right? You've just kind of got diffusion. But you could always do it on, it depends what the question is, right? You can always use a diffusion model and then compare it to that on a small regions. And, yeah.
Okay, thank you very much.
Hi, elegant work, beautiful work. One of the things that I wasn't entirely clear about is when you said that you're taking different sections, different stains, so that the same stain is separated by, was it 350 microns? Mm-hmm. So if I understand that right, then you get very small resolution in plane and a big fat distance between planes.
Yeah.
So that's very similar to what Thomas has been dealing with for a long time with the chemoarchitectural problem. How are you dealing with the interpolation across voxels that are so anisotropic?
So here we just, we mostly do the tractography with isotropic resolution, and we just go down to 0.4. So you have one microscopy slide within that, and you have to think that that is representative of the whole voxel. What we can do is that we can combine information potentially across the different microscopy slides, because all of them can tell you about fiber orientations, as the structure tensor and the PLI. And so then you would have a more dense sampling, but then you're mixing information from different sources, and there's a bit of a messiness to that in some ways. So that's not what we've shown today, but it's possible to do that.
Well, you've had you've done the same sort of thing with the chemoarchitecture.
Yeah. I think that's very different. Neuroimaging has that really
Anyway, it's lovely stuff.
Cool. Any more questions for now? Okay, we love the questions, so thank you. Just to quickly say, all of our data is openly available, so please get in touch if you'd like a copy. We do have to ask for a data sharing agreement, just to make sure that it's used for ethical and research purposes. And we also have some online information and tutorials and stuff to get people started. I'm going to say all of my thanks to everyone before we move on, because this is obviously a huge team effort. But really today what we wanted to do was have an interactive workshop. So we hope if you've got laptops, please get them out. There is a kind of web browser based workspace that you can explore some of the data, run some MRI correlations and do some registrations and run hybrid tractography and have an explore of whatever you think is really interesting. We do ask that the data doesn't leave the servers, so please don't try to download it outside of that workspace. I will put these back up afterwards, but maybe we can just give you a quick tour of what it's going to look like if my thing is still open. Let's see. Yeah, okay. So you should get a Jupyter Lab environment. So when you first load up, if you go into the terminal, oh no, not the terminal. What am I doing today? We need to go into the other one. Sorry, we're a bit new to this system as well. So you can get a desktop, and the notebooks are gonna be running stuff in the notebooks, and then opening FSL, having a look at the data, blah, 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 that kind of stuff. So it will be a bit of moving between a desktop, which will open here. Oh, connection closed. It's not good. But you'll get a desktop environment. And then down the side here, there's 3 different notebooks. So one is called Exploring the Data. We've given you a subset of the data to just check out, see what you think. We've given some there's all of it. We've given some ideas of things that you might be interested to look at, but please, however you would like to. The only thing is that we can't load the SBS files. Are you going to sort it out for me? Thank you. So we can't load the SBS files on this system just because they're 5 gigabytes each, but we've got 3 laptops at the back there that have some of the data for you as well. So there's this exploring one, and then there's a second notebook where you can run co-registrations and run some correlations and things. And then a third one, which is all about hybrid tractography, given the time limits, like do whatever you like. Please ask us lots of questions. We would love that. If you want to copy and paste between the domains, it's best to, between the desktop and this, just open a text file, copy into the text file, it'll move across and copy it out again. It's a little bit fiddly. But we can come around and help out with this. It's probably a bit easier than me doing it this way. So I will put these back up on the screen, and hopefully people can log in. And yeah, ask us lots of things, please. Cool. Good luck.